I ran into this article in Nature today, and I wanted to talk about it because it mentions an ongoing study that I think is really good and will hopefully answer a lot of the questions that many of us have. So this article was COVID reinfections are unusual but could still help the virus to spread. Large study of UK health workers suggests that most people are immune for months after catching COVID-19. Okay, so we're going through here and it says they're talking about this study called the SARS-CoV-2 Immunity and Reinfection Evaluation Study. So when I saw this, I tried to find the paper that they're talking about, but there is no paper. I think this is just information from a press release. And so I went and Googled this study, and I found their website. And this is their website, the Siren study. And this is their website to enroll people. And then it's just a very simple one-page website, and there's this participant information leaflet. So this is the participant information leaflet. So I try to figure out what is this study doing. Okay, so it was pretty helpful. So they talk about, okay, what is this? What are they, why are they doing this study? And so what they're going to do is they're going to have people volunteer and they're going to swab their nasopharynx and draw blood regularly over time to be able to assess whether prior infection measured with the antibody test that's in the blood protects against future infection measured through the detection of a virus on a swab test. I assume this is going to be a PCR test. So these are their goals. The blood test will allow us to understand the number of healthcare workers infected by COVID in the last few months and allow us to understand whether there are differences related to age, ethnicity, and other factors. So all the people that participate in the study, they're going to get a baseline blood test that tells them whether or not they've been infected, and they'll measure them throughout the time course of this study, and they'll see if there's any differences related to age, ethnicities, and gender, other factors. So that's a pretty important thing to know, which we don't really have a great handle on in a prospective study yet. We have a lot of retrospective data on it. By taking regular samples, we can measure what proportion of frontline NHS staff are exposed to SARS-CoV-2 and improve our understanding of how quickly it spreads over the coming months. So also looking at healthcare workers, which are probably the highest exposure group, or one of the highest exposure groups. By taking blood samples, we understand how individuals' antibodies levels change over time and the different types of antibodies that may be present. This is great because this is exactly the kind of information we want to know. We want to know how quickly do the antibody levels disappear, and there's a lot of studies that look at that, but this will be a very systematic way of looking at it, where people are sampled on a very regular basis instead of doing it retrospectively, which most of the papers so far have been doing. Then they will investigate how viruses from different individuals relate to each other by comparing the genetics. So they're going to sequence these viruses and see any genetic changes that over time and over geography. That'll be great. Where participants are swab positive several times, we will seek to understand how infectious they may be through attempting to grow the virus from their samples and explore the changes in genetic material. So if they get reinfected, they're going to culture and sequence these things. And then if individuals are admitted to the hospital, they will explore how. So they want to figure out what factors affect severity of disease. So these are all like the important questions that everyone wants to know, and this study is trying to answer them, which I think is great. So what they're going to do, they say, it says follow-up will last for 12 months, and the blood and swab tests will happen regularly. For most people, they're going to get questionnaires and blood and swab tests every 2 weeks. So that is a lot of sampling. They're going to sample everyone's blood and nasopharynx every 2 weeks for 12 months. So this is going to be a really huge data set. All right, so now we kind of know what this study is, and I'm going to read this article. And so basically, I hate reading these articles because I would rather just read the paper, because a lot of times what they say in these articles, trying to summarize the paper, they leave out important details or they kind of misinterpret what they're saying. But they say reduce the risk of catching infection by 83% for 5 months if you've had it before. So later on, I think you can figure out how they got to that number. So data suggests that repeated reinfections are rare. They occurred in fewer than 1% of 6,600 participants. So I guess what happened is when they got all their participants, about 6,600 already had antibodies to it at the start of the study. And then throughout the course of the study, about 1% of them got reinfected. So reinfection is possible, but it seems to be very low, 1%. Of course, that percentage is going to get higher as the surveillance time frame increases. So they've been doing this study for about 5 months, and they found 44 possible reinfections. And then in the group, so looks like they have about 20,600. So they have 6,600 that had antibodies at the beginning of the study and 14,000 that did not. Out of those people, 318 tested positive, and out of this group, 44 got reinfection. So if you do the math on that, that becomes the 83% protection. So this is very similar to the vaccine studies where, people with vaccines in the Moderna study had or I think in the Pfizer study had 9 infections, and then in the non-vaccine placebo group, they had 169. And so if you say that we expect 169, but we only got 9, that's 95%. So in this case, the previous infection is 83% protective. So very similar to the vaccine. Um, so some of the reinfections are still being evaluated. All 44 are considered possible reinfections. So far, 2 of these have classified as probable. I'm guessing what happened here, and see with the full study, we'll know. These 44 retested positive with PCR. These 2, they sequenced the virus or they grew the virus in culture, so they know that it's viable virus. The PCR test sometimes can catch non-viable virus. It may not be a true reinfection. The study did not assess whether symptoms were better or worse during the second infections than the first, but Hopkins notes that only about 30% of the people with the possible reinfections report any symptoms. That's kind of important, because if 30% of these possible reinfections were actual reinfections, that means these people were having asymptomatic infections, which means that those are the people that in the vaccine studies would not have been captured, because remember, the vaccine studies only looked for symptomatic reinfections. So if people are getting asymptomatic reinfections after being infected with the virus, then it kind of makes sense that you could have asymptomatic reinfections with the vaccine as well, because they're probably similar in terms of immunity. But so there's a lot of unknowns, because this is very preliminary. They finished 5 months of this 12-month study, but I think this will be a very important data set to answer a lot of the questions that we have, you know, and also the new UK variant is going through the UK now, and we'll see if that variant shows up more often as a reinfection. That could give you a clue on whether infection with the previous strain, the original strain, would give you protection against the new variant, or if it's different. And then probably the South African other variants are going to go through the UK, and that'll show up probably in this study as well. So this will answer a lot of the questions, and how long does the antibodies last? How long is it protective for? Do the reinfections go up after a certain length of time? These are all the questions that are important, and that this study will probably answer. So I hope they put out an interim paper kind of giving the 5-month data with the method, so that we can reevaluate it better. But when it comes out, it'll answer a lot of the questions. This is a similar study that has been published with a much smaller data set, but is pretty similar. They looked at 136 healthcare workers that had been infected. 76 of them had mild or asymptomatic infections that were picked up just by serial sampling. So these people did not have symptoms, but they were found to have had asymptomatic or mild disease. And then what they found out, that even despite that half the number in their cohort had asymptomatic or mild disease, most of them still had neutralizing antibodies. And then they found out that T cell responses tended to be lower following asymptomatic infection, while neutralizing antibody titers were maintained even if they had asymptomatic disease. So antibody titers and T cell responses are higher with symptomatic disease, but neutralizing antibody titers are the same even in asymptomatic disease. So they said sometimes the T cell responses and antibody responses were discordant. So some people had good T cell responses with relatively poor antibody responses, and vice versa. And then 11% had lacked any neutralizing antibodies and had undetectable T cell responses to the spike protein, but they had T cell responses to some of the other antigens. So their conclusion is our findings suggest that the majority of individuals with mild or asymptomatic SARS-CoV-2 infection carry neutralizing antibodies complemented by multispecific T cell responses in the time period that they looked at, which was 6 to 18 weeks after mild or asymptomatic SARS-CoV-2 infection. So I just wanted to kind of go over the first two figures because I think they're kind of the most important in this study. Let's see, opening a new tab. All right. So let's see. This is ELISpot data. Let's read. Yeah, T cells responses. So this is ELISpot. So the way this works is they get the white blood cells and the lymphocytes from the peripheral blood of these patients, and then they put them on a target, which is like the spike protein or the nucleocapsid protein, and when T cells recognize the antigen, they produce interferon gamma, and the interferon gamma can be picked up with an ELISA, so you get an ELISpot. So you see that there's more ELISpot against the nucleocapsid than the spike protein. Then these are called, these are like epitope peptides. So these are specific using peptides instead of the whole protein. So same thing there. This part right here is comparing laboratory confirmed SARS-CoV-2 to people that tested negative by PCR test but had some symptoms. These are the case-defining symptoms, and they use case-defining symptoms as cough, fever, shortness of breath, or loss of taste and smell. And then this is asymptomatic, and this is a group of blood samples they had before the thing started. So these people definitely had not been exposed to the virus whatsoever. And then the black means that the darker means they had more reactivity. So you can see, obviously, the people that have the actual tested positive have much higher reactivity than everyone else. It's a little, and obviously there's some kind of cross reactivity because there is some reactivity in people even before the virus even existed. So there's some cross reactivity to the epitopes there and some mild cross reactivity amongst asymptomatic people. And people who probably didn't have any reaction to the virus. Then this is the neutralizing antibody data. So you see here, this is this is antibodies to the spike protein, antibodies to the nucleocapsid protein. So you see there's a few people that have zero responses. Their titers are essentially zero. And then this is the neutralizing antibody titers. And you see there's a few people that have no neutralizing antibody. And the neutralizing antibody study is done. They put the serum in with a live culture of the virus, and they see what titer of the serum do you need to reduce the number of infections, so viral infections in the culture, down by 50%. So these people you could put on full strength, and it didn't reduce the virus, so it doesn't neutralize the virus. Neutralizing antibodies stop the virus from being able to infect cells. And so this is the proportion of the people in their group. So most of them had high titers, and then low, and then a few had none. And then this is the correlation between the spike protein and the neutralization antibodies. And there's a kind of a weak relationship between the spike protein titers and the neutralization antibody titers, and basically no relationship. It's flat. And then looking here, people that had case-defined COVID and then non-case-defined COVID and asymptomatic COVID. So as you become less symptomatic, the titers of the spike protein antibody go down, but the titers of the neutralizing antibody stays pretty constant. So asymptomatic people have less antibody response to the spike protein, but in terms of being able to neutralize the virus, it's about the same. And then this I thought at first was a little counterintuitive, but they looked at antibody titers by age, and it looked like older people had higher titers, which doesn't make sense initially because older people's immune systems are not as robust, and there is some senescence to the immune response, and so you would expect them to have lower titers of the neutralizing antibody, but older people had higher. And then when I saw this, it kind of made a little more sense because what you'll notice is that the people that have almost no titers are usually the younger people, and these people probably had less severe disease. Is my take on it. Because we know that asymptomatic and milder disease is more common in younger people and less common in older people. So the take home is between these two studies, and I'm waiting for the final paper of the Siren study to come out, is that looks like past infection is about as good as the vaccine. It's 83% protective, lasts for at least for 5 months. That's what we know about the vaccine, lasts for about 4 months. And that even asymptomatic disease gives you neutralizing antibodies and T cell responses. So that's overall pretty good news. It will be very interesting to see how the new variants affect the study as this goes forward and see if it's protective to the new variants. So if you have any questions, comment below, and thanks for watching.